Okay, let's resume from where we left in the first part of the lecture. This is the product of transcription and the post-transcriptional modification. It's a messenger RNA or mRNA which is now ready to convey that information in the protein coding sequence in the ribosome to produce a protein. This is what is going to be done in the process we're talking right now, translation, where the mRNA enters the ribosomes and the information get read and forms the protein. So how is this done? Well, this is done following a language, what is called a genetic code, which is shared by all living organisms. This is a language which is based on 3 nucleotides words referred to as codons, where you have that each of these codons, this sequence of 3 nucleotides, correspond to only one specific amino acid. So now the ribosome can go through these mRNA, read 3 nucleotides at a time, and for what they can read, for the codon that they can see, like in this example here, they can sort out which amino acid they can add, like in this case with methionine, alanine, and tyrosine. This picture here summarizes exactly what the genetic code is. This shows you the 64 possible combinations of these 3 letters, the 3 nucleotides, and the amino acid that they correspond to. We'll discuss in more detail in the next slides. So what are the features of the genetic code? Well, it has very specific features. As I said before, it's shared by all living organisms, so it's universal. There are some slight changes in the way mitochondria read the mRNA, but basically it's shared by all living organisms. It's universal, first thing. Second thing is commaless. These codons that I mentioned, they're not separated by any nucleotides. They are one right after each other, like you see here on the right side here. There are no nucleotides in between, no 1 or 2, like in this example. Other thing is non-overlapping. So these codons are just one after the other. They do not share any nucleotide, like in this example here. So one right after, they don't share anything. There's nothing in between. The other thing, they are read only in one specific reading frame. If you think these codons are made by 3 nucleotides, theoretically you could have 3 different starting points or 3 different ways you can read the same RNA. For example, here you could have that you can start from this AUG, which is how it would start in reality, or you could start from UGG, the U here. So the first codon could be UGG, or if you start from this G, now you would start with GGC here. So the third codon here, and so all of them will give origin to obviously different sequences of amino acid. Now, the genetic code ensures that every mRNA can be read only in one specific way. You'll see later on how it does that. Now, it's also one last property of the genetic code is what is called degenerate. What it means is that since it has a lot of possible combination with 4 nucleotides or 3 nucleotides with 4 possible choice for each nucleotide, you can come out with 64 combination, which is what you see right here. Now, there are only 20 amino acids. What it means is that now you may have more codons than the amino acid. And in fact, you have more codons. So many amino acids are actually coded by 2 to 6 different codons. So that's what it means degenerate. You may have multiple codons for a single amino acid. This is true for all amino acids except for methionine, which is, you'll see is always the first one, and tryptophan. Now, all these 64 codons that you can see here in the genetic code, now you have the 61 indeed they give origin to amino acids. One of these 61 is always methionine and is actually always used as the start codon. That's what really ensures that one mRNA is always read in the same reading frame. So what ribosomes do, they actually look for this first codon to know exactly where they have to start the translation process. So that's the AUG codon here corresponding to methionine. That's the first one in all mRNA. And then there are also 3 codons here highlighted in red which do not correspond to any amino acids. And in fact, what they're used for is actually to terminate, to stop translation in the process. You'll see how it works when we go through the specific process itself. So, based on what I say, all the properties that I told you, how would you translate an mRNA like this one? So, it's always read in a specific reading frame. And what ensures that is read in a specific frame is by starting always with a methionine. So the first codon will always be an AUG. Okay, and that's exactly what the ribosome do. They scan this messenger RNA, look for the first methionine codon here, and this is where they will be starting. And so ahead of this first codon, this region will be untranslated, will correspond to the 5' untranslated region. And then you see later on there is also a 3'. And then from there on, they will read one codon at a time in a commonless, non-overlapping way. So the next codon will be the CCG here, corresponding to proline. There will be CCG right here. The next one will be GGC. These will correspond to glycine. So all of these amino acids will be joined together at the ribosome. Then there will be isoleucine here. And then finally, they will encounter this codon, which is one of the 3 possible stop codons, and translation will stop there. There will be no amino acid introduced there, so this will be a tetrapeptide, and then this region here will be what will correspond to the 3' untranslated region. Again, this will be read starting from methionine, from the first AUG, and then this will be read in a commonless, non-overlapping way, one codon at a time, until a stop codon will be encountered. Now, this is just a summary, a few overview of translation. So it's a mRNA-directed polypeptide synthesis is always done in the cytoplasm, both in eukaryotes and in prokaryotes. So how is it done? The mRNA is associated with the ribosome, which then can read it. And they read it one codon at a time in the direction going from the 5' to the 3', always unidirectional reading. Now, the amino acids, how each codon is translated to amino acid is because there are transfer RNAs, other type of RNA molecules that actually are the one that bring the specific amino acid for the codon to the ribosome. We'll see how they do that. And then for the amino acid, all the amino acid will be joined one at a time through a peptide bond, forming ultimately the polypeptide chain, which will always be growing from the N terminus. So you always have an half amino group free at the beginning, and will end with a C terminus, so a alpha carboxy group free at the end of the polypeptide. As I said before, the amino acids are brought to the ribosome using specific transfer RNA molecules. What I mean is that for each amino acid, there is one tRNA with its own specific RNA sequence. They don't share, so different amino acids do not share the same tRNA. These RNA molecules are short RNA molecules from 75 to 90 nucleotides long, okay, and they have areas where they actually are complementary with each other, where they form partial double helix. So in fact, they have a very complex 3-dimensional structure. Now, of this part of the tRNA, all the tRNA, no matter which amino acid they are associated with, they have 2 key features. They have one region here, one of these loops, which is referred to as the anticodon. The anticodon is always read in the 5' to 3' direction. So if you look back, going back here, this is the 5', it's covered here, so it will be the 5' this way. So it is always read 5' to 3'. So this is the first codon, the first nucleotide, second and third. So an anticodon is that sequence of 3 nucleotides that are complementary to the codon that codes for the amino acid, which now is joined in the same tRNA at the 3' end. So in this region right here. So by doing that, now you have an anticodon that's specific for a codon which corresponds to this amino acid. And therefore, by doing that, this anticodon, which is brought to the mRNA through the ribosomes, now can bring a specific amino acid for a specific codon which is present in there. For example, here the tRNA for serine, so it would have serine attached here, and now you would have an anticodon here, which is UCA read, so you can read it. In this case, is red, just to show you the complementarity. UCA, so the reverse, but this will be the first one, and will correspond to a codon AGU, so complementary to this sequence right here. And by doing that, basically we'll be bringing serine for this codon, which if you go back to the genetic code, indeed correspond to serine. Now, as we said before, the genetic code is degenerate, so there might be more than one codon for one specific amino acid, up to 6 different codons. One way in which this is done is also by the tRNA wobble. What does it mean? What it means is that in reality, the anticodon of the tRNA, because of the angle of these 3 nucleotides, one after the other, they're not perfectly straight. You have that the first nucleotide in the anticodon, again, you read the 5' to 3', so this will be the first one, may actually associate or form hydrogen bondings with more than one nucleotide in the codon. So, for example, here you see GAG, the same one here, can actually recognize this first nucleotide here, can actually pair with cytosine, which is what you would expect, but can actually pair with uracil as well. And this is because, again, they're not perfectly on the same plane. So this hydrogen bond is fairly loose in this case. The other 2 nucleotides, so 2 and 3 in the anticodon, instead they always have a perfect pairing, normal pairing. Okay. So now, depending on what this nucleotide is, you may have different possible pairing. So G, as we said before, can match with either C or U, cytosine or uracil. If you have a uracil here now, you can associate it either with adenine, which is what you would expect, but also with guanosine. And then, finally, there is also a modified nucleotide, which maybe enter as the first nucleotide in some tRNA, which actually can form bonding with 3 different nucleotides. So cytosine, adenine, and uracil. So what it means is, for example, if you have a GAI here, now these could actually match with CUC, CUA, or CUU. So the same anticodon at this point could actually bring whatever amino acid is associated with that to 3 different codons. Now, these amino acids are attached to their specific tRNA through a process called amino acylation or charging. You can remember charging of the tRNA or amino acylation, but you can use either one. This is done by a specific enzyme, amino acyl tRNA synthases. There is one for each amino acid, and this is the enzyme that will recognize the specific tRNA with the specific anticodon and would attach the specific amino acid to that tRNA. This is the very first step in translation. And now, moving on here. Second key component, so the tRNA are essential to bring the amino acid to the ribosome. Let's talk about the ribosomes. What are the ribosomes? The ribosomes are actually structures which are made as a complex of RNA and proteins together. This combination of RNA and proteins form 2 different subunits, one referred to as the small subunit and one referred to as the large. When translation is not happening, these 2 subunits are separate from each other. When translation is happening, they get together and they form 3 sites, which are essential for the translation process. There is an A site, which stands for amino acyl site. This is where the tRNA carrying the amino acid is brought into the ribosomes. Then you have a peptidyl site or P site. This is where actually the transferring of the growing peptide to the new amino acid takes place. We'll see every single step here. And then finally there's a third site called the E site, the exit site, which is where the tRNA, once it has given its amino acid, is released without uncharged, completely uncharged, so without the amino acid. This is exactly what I was saying. So this is the structure of the full ribosome. So you would have a large subunit and a small subunit. Both of them are made by RNA and proteins combined together. And when they're joined together, you have the three sites that are formed. A amino acyl site, that's where the tRNA will be brought in. Peptidyl site, which is where the growing peptide gets transferred to the new amino acid, and then exit site, which is where the uncharged tRNA gets released. Let's go through the steps of translation. Like in transcription, translation can be divided into three different steps. First one called initiation, that's exactly the same names. Initiation, this is where all the machinery is brought together, exactly like in transcription. If you remember transcription, initiation in transcription, that's when the RNA polymerase is brought to the promoter so that then it can start copying. So everything is brought together in this case to an initiation. The RNA, the ribosome, and so on. Then elongation, that's exactly where the amino acids are joined together. The codons are read and the corresponding amino acids are joined together through the formation of peptide bonds. And then termination, it's when the process is finished, all the different components, the mRNA, final tRNA, the ribosome, all gets released, the two subunits, and translation is done. Let's go one step at a time. Initiation. Again, this is the process where all the necessary components for translation are brought together. The necessary components are obviously the ribosome, the messenger RNA, and the first tRNA corresponding to methionine. You remember, translation always starts with an AUG. That's the first colon is read. So how does it work? Well, the first step is that the small subunit of the ribosome is combined with methionine and GTP. So methionine tRNA. So this will be the tRNA with the anticodon recognizing the AUG and methionine attached to it. This is joined to the small subunit and with GTP. Now, this complex here then joins together with the mRNA, and these mRNAs start being scanned until the AUG, the first codon, the start codon, is found. This is done, the recognition is done thanks to the anticodon present in the methionine tRNA here. At the same time, the large subunit is also brought in. GTP is broken down in GDP and inorganic phosphate, releasing energy for this process to help. Once this is done, now you have this, sorry, this was actually supposed to be on top, this picture, they're reversed. Once this is done, now you have that the full ribosomes with the three sites here. The tRNA for methionine is aligned to the start codon, the AUG, and the mRNA. And now you're ready to perform translation. So the first amino acid is brought in place. It's aligned with the start codon. Translation or elongation can start. How is elongation happening? There is a lot of things happening. This is a big blah blah blah. Let's move on straight to the picture. It's much more direct than all those words. So this is the situation after initiation. You have that the tRNA for methionine is in the peptidyl site. It's aligned with the start codon, right? And now you have the second codon is now instead in the amino acyl site. This site right here. This is the second codon. The others are outside. So the very first step in elongation now is they bring there is the second tRNA, the one corresponding to the second codon, is brought in the amino acyl site here. This is brought in thanks to the help of additional energy coming from GTP that's broken down into GDP and inorganic phosphate. So the second amino acid is brought in there in the amino acyl tRNA. Now you have these two amino acids side by side, still separate from each other. In the next step, now there is an enzyme, present in the P site here, which is referred to as the peptidyl transferase enzyme, which in the peptidyl site, what it does, it transfers the first amino acid, which will be methionine, from its tRNA to the second amino acid. So it's forming the first peptide bond. Now you start having a dipeptide attached to the second tRNA. Once this has happened, now the next step is the mRNA, or the ribosome, I should say, moves compared to the RNA by 1 codon. This is done again with other energy being entered by breaking down another molecule of GTP into GDP and inorganic phosphate. So now you have that this ribosome moved by 1 codon. So what happens now is that the uncharged tRNA, first tRNA that had methionine, now is uncharged because methionine has been transferred, and find itself from the peptidyl site move to the exit site. Whereas the second tRNA that now has the dipeptide, methionine, and the second amino acid find itself into the peptidyl site. When this happens, now you have that the first tRNA gets released, the uncharged one gets released, and now you're back to square one, to the same position that you were before. But in this case, now you would have the second tRNA here with the 2 amino acid attached to it. What's going to happen next? Well, you have the third. Now you would have the third tRNA being brought in with the third. You would have now the third codon in the amino acyl site, and so the third tRNA is brought in with the help of energy again, and now you would have peptidyl transfer would transfer the dipeptide to the third amino acid, and this will keep going and going and going cyclically. So it's the same step with one peptide bond formed in each cycle, with the growing peptide being transferred to the new tRNA all the time, and this leading to the switch or the movement of the ribosome from one codon to the next time for each cycle. By doing that, all the codons will be read until termination happens, and termination happens when in the amino acyl site you end up having one of the 3 stop codons. UAG, UAA, or UGA enters here. When this enters here, there is no tRNA carrying any amino acid anymore here. Instead, there is another molecule called a release factor or termination factor, which enters the site, and when it does that, it stimulates finally peptidyl transfer cleavage here. So the cutting basically of the bond between the peptide and the last tRNA, or the tRNA that had carried the last amino acid here. So by doing that, now you have that the peptide or polypeptide is finally released from the ribosome, and this also leads to the separation of all the components. So the large subunit will separate from the small subunits, the RNA, messenger RNA gets released, same thing for the tRNA, uncharged tRNA, and the release factor or termination factor will be released as well. At this point, translation is done. The polypeptide has been formed and is done. Now, this process that I went through with you, it doesn't take place, oftentimes doesn't take place once, one ribosome at a time. It's very common to see messenger RNA, both in eukaryotes and in prokaryotes, to be read by multiple ribosomes at the time. So look at this. This will be the same messenger RNA being read by multiple ribosomes at the time. So by doing that, now you have this one just started and this one is basically done. So you have multiple proteins being formed much faster than if one ribosome was allowed to go through that at the time. By doing that, you can increase the rate of polypeptide synthesis for each messenger RNA. Keep in mind that each messenger RNA may last for just few minutes or a few hours, some of them even a few seconds. They don't last very long. It depends how much protein they have to produce. So in that period of time, you want to produce as much protein as possible. The formation of polysomes, which is the structure you see right here, having multiple ribosomes bound to the same mRNA, leads to do that in the fastest way possible. In prokaryotes, actually differently from eukaryotes, now you can have also that the transcription and translation can be done at the same time. The reason why this is possible in prokaryotes and not in eukaryotes is for two main reasons. The first one is that prokaryotes do not have introns. So as soon as the RNA is done, it's a mature messenger RNA ready to be translated. So the ribosome can immediately start binding to it and start producing proteins. The second reason is that in prokaryotes, unlike in eukaryotes, the mRNA doesn't have to be transferred from the nucleus to the cytoplasm because there is no nucleus in the prokaryotes. And so the ribosome have direct access basically to the genome, and the RNA just newly synthesized by the genome. This picture, this electron microscopy picture exactly shows you that. This will be the genomic DNA, the chromosome of the bacterium, and these are messenger RNA that are formed right now at the moment. At the same time, they're already bound by different ribosomes which are already translating them. Now, the proteins that is formed, they can be the final product, or they may undergo some different processing and conversion into before they actually become the final functional form. Now, there are different types of modification that can take place. There can be removal of amino acids or addition of different other molecules like lipids or carbohydrates, which would take place in the endoplasmic reticulum, if you recall. There are proteins, chaperone proteins, where we talk about that when we talk about proteins that help with the folding of the proteins oftentimes. And these, some proteins also are synthesized first as an inactive form, which might stay like that until they either get exposed to conditions that can convert it into the active form, such for example, some enzymes in the stomach, they're produced inactive, but then when they're exposed to the low pH or high proton concentration, they get activated. Or sometimes they need to be actually cleaved, they need to be cut. Some amino acid sequence that have to be removed for the protein actually to become active is very common, for example, among the factors that cause clotting in your blood. The other thing is that many proteins that are synthesized also ultimately they may be synthesized in the cytoplasm, but they have to function in different organelles, and usually this is done through the presence. So when they're synthesized, they would have a sequence at the very beginning, a signal sequence, which tells other proteins where this polypeptide has to be shuffled to. For example, there are nuclear localizing signal sequence of few amino acids present in proteins that are synthesized in the cytoplasm, but they have to work in the nucleus. There are mitochondrial localizing sequences as well for other proteins that are done in the cytoplasm and have to end up in the mitochondria. Once these proteins they enter the final organelles, usually the signal peptides in the N terminals here gets removed, gets cleaved, and then they can finally do their function in their final organelles. Last thing I'm going to talk about in the second part of this lecture is how mutations affect proteins. Mutations, we refer as mutation any change in the nucleotide sequence of genetic material. As we said before, genes correspond to 1 to 2% of the total genome. So you can see how many of these changes of these mutations might not affect genes and therefore might not affect the protein codes by the genes. However, some of these mutations they are in the protein coding sequence. And so they can have different effects depending on what they really do. In some case, some mutation, they might just change a codon, like imagine this case. So this is the normal sequence of a gene. You may have one mutation where you have one codon that get changed. So in this case, this thymine would have been changed into cytosine, and therefore this adenine into guanine. Single change point mutation, that's what it's called. However, the change of this codon because of the degeneracy of the genetic code doesn't cause a change in the amino acid. Both this codon and this codon right here code for glycine. So the peptide, the protein formed by this gene, is not altered by this mutation. This mutation is referred to as a silent mutation. In other case, this change may actually lead to a change in function. So here you can have a change, for example, in this serine here. So this codon here, TCA, gets changed into CCA. And so by doing that, now you have that serine is replaced by proline, which is a very different type of amino acid. This would have an effect on the function of the protein. So in this case, you're referring to as missense mutations. Sometimes you may have, though the amino acid might be changed as well, but the amino acid that is changed into has very similar property than the original amino acid. And so in this case, this mutation, even though it changes the amino acid, it might not have, it will be a missense mutation, but it still would not have a functional effect on the polypeptide. Other type of mutation you can have is that these might cause the introduction, the change of one codon from an amino acid coding codon, doesn't sound very good, to a stop codon. So for example, in this example here, so this codon here, ACG, has been changed to ACT right here, which basically was introduced a UGA, which is a stop codon. This obviously will lead to a truncated form of the protein. So now the ribosome are reading there. Once they reach the stop codon, they don't introduce any more amino acid. They stop right there. So the protein here, which could have maybe be made of hundreds of amino acids, now is maybe only by 4, and obviously would have lost its function. Other mutations, devastating mutation in this case, usually are what we call frame shift mutations. These frame shift mutations, what they do, they consist on the insertion or removal. Here it talks about insertion, but also insertion of removal of one or two or multiple of 1 and 2 nucleotides. By doing that, so now basically what it does, it completely changes what is called the reading frame of the mRNA. Do you remember, each RNA has to be read in a very specific sequence of codons. But now if you introduce, for example, like in this case, 1 nucleotide or 2, now you get that all the codons from that point on are gonna be changed, right? So the reading frame from there on is being shifted. So this is called a frame shift type of mutation. The same effect you would have if you have 2 nucleotides introduced. If you have 3 or multiple of 3, obviously at this point you will be inserting a new amino acid, but the remaining reading frame will stay the same. Similar effect you have if you remove 1 or 2 nucleotides, you would have a frame shift mutation. But if you remove 3 or multiples of 3, you would have loss of 1 or 2 or 3 amino acids, depending whether it's 3 or multiple of 3s. But then from that point on, the reading frame will still stay the same. So this will lead to the loss of 1 or 2 amino acids, or but not change in reading frame. So in summary, what did we talk about in the first and second half of these recordings here? What we talked about is, first of all, we went through what were the discoveries that led to understand that there is a connection between DNA or genetic material and proteins. Then we talked about the central dogma. So that dogma that explains how the information flows from the DNA first to messenger RNA to transcription, and then to proteins to translation. And then we went through the two processes, transcription and translations. For transcription, we first went through the gene structure, the transcriptional unit made of promoters, RNA coding sequence, and then the termination sequence. And then we talk about the different step of how transcription is carried. Then we talked about how the precursors mRNA in eukaryotes is converted into the final messenger RNA through 3 additional steps referred to as post-transcriptional modification, consisting of a 5' capping with guanine, 3' polyadenylation, and then the removal of introns through splicing. And then we talked about how the removal of intron can be different depending on tissues through the process of alternative splicing. Then we move to translation. Before talking about the steps, we talked about the genetic code, that language that is used by all living organisms in the same way to read mRNAs, based on codons and anticodons in tRNA. And then we went through the steps, how the steps are carried over, initiation, elongation, and termination as well. Finally, we talked about the post-translational modification of proteins, how they can be changed or turned active, or how they can be delivered also into different organelles. And finally, we talk about the effect of different type of mutation in proteins.